So we'd now like to welcome Paul Magwene from Duke University.
Great, good morning. So today I want to tell you about a technique my lab has developed for looking at single-cell heterogeneity in division rates and mortality in yeast. And the motivation for doing this is several-fold. We were interested in studying responses to cellular stress at the single-cell level, but we're also microbial evolutionary biologists, and we're interested in developing single-cell measures of fitness. The work I'm going to tell you about today is focused on looking at yeast responses to thermal stress. So we'll be looking at the heat stress response. And I want to say that all the work I'm presenting is the work of a very talented graduate student who recently defended his PhD, Colin Maxwell. This method I'm going to tell you about was his brainchild, and the results represent his hard work. So I think it's fair to say that bulk population growth rate is probably the most measured variable in all of microbiology. And there's many good reasons for that. It's easy to measure, and it provides a good indicator of how cellular physiology changes in response to changes in the growth environment. If you're a microbial ecologist or evolutionary biologist, it's a good proxy for fitness. However, a simple reliance on bulk population growth rate hides a lot of details that we're interested in. For example, if you see a shift in bulk population growth rate, you can't tell whether that's due to a change in fecundity, the number of new individuals produced per unit time, or a change in mortality, or some combination of the two. Simply focusing on bulk population growth rate also doesn't allow you to characterize heterogeneity in populations. We know genetically homogeneous populations often produce biologically interesting heterogeneity, such as the fact that antibiotic-resistant persister cells in bacterial populations are preferentially coming from a subdistribution of cells that are growing slowly. So we need single-cell techniques to be able to distinguish such phenomena. And the technique I'm going to tell you about today is simple, inexpensive, and doesn't require any fancy equipment other than having access to a decent fluorescence microscope that can acquire Z-stacks. And what we're going to do, as I sort of alluded to in my title slide, is we're going to exploit the fact that yeast keep a record of their reproductive output on their cell wall in the form of bud scars. The technique we've developed is very simple. It's called TrackScar, and it's essentially a molecular mark release recapture technique. And the way we're going to measure single-cell growth rates is illustrated here. Let's see if I can get the mouse going. At some initial time 0, we'll call T0, we're going to label bud scars in a population of cells with a bud scar stain like wheat germ agglutinin coupled to a fluorophore, our favorite fluorophore, in this case a cyan fluorophore. We then wash out the dye, put the cell population into the growth conditions of interest for some fixed time interval. And then after that, at the end of that time interval, at some time T1, we're going to restain that population of cells, this time with a second color. We then image several hundred cells from that population, and we count the number of bud scars that are only stained yellow, conditional on at least a single, having at least a single bud scar that was stained with the initial stain. So what this gives us then is the number of new bud scars in that time interval of interest. And it's a very unambiguous, very straightforward measure. You can count directly on image cells, as is shown here at the bottom. We've got cells with 3, 6, and 7 bud scars in this time interval. Okay. So this is the simplest version of this TrackScar technique, and it directly gives us division rates or reproductive output per unit time. Well, the way we most often analyze and think about this data is in the form of these frequency polygons, as shown here. What we have is a distribution showing the number of divisions individual cells have gone through in this case, a 6-hour time interval. This is the strain S288C under happy conditions. And we see in this case, most cells produce 5 or 6 daughters in the 6 hours. So roughly 85% of the population is dividing 5 or 6 times. And then there's a small percentage that are growing, dividing 4 or 7 times. Okay. So this very discrete measure on individual cells, then we can summarize sort of the population behavior, but still retaining this richness. So what we're going to look at for the rest of the talk is how these patterns of fecundity, and I'll also get to mortality, change as we expose cells to heat stress. And I'm going to focus on 3 strains that we've examined. This is part of a larger study where we looked at thermal sensitivity in a collection of 100 different Saccharomyces cerevisiae backgrounds. The 3 strains I'm going to focus on are S288C, shown in red, just as sort of a reference. And then 2 strains that we're going to call sensitive in that they show decreases in bulk population growth rate. This is a bulk measure I'm showing you here. Bulk population growth rate at 35.5 degrees Celsius relative to 30 degrees Celsius. So they grow slightly worse at this sort of very intermediate temperature. So here's the S288C data just for reference. This is the robust response. And we see that at these 2 temperatures, it grows just fine. In fact, S288C grows a little bit better at 35.5 degrees Celsius than at 30 degrees Celsius. Very narrow fecundity distributions. It's just cranking along. By contrast, if we look at these 2 sensitive strains, we start to see interesting behavior now that we can look at this at the single cell level. So for example, on the left is the strain that we previously labeled as blue in this slide here. We'll call this the moderately sensitive strain. At 35.5 degrees Celsius, the mode of this distribution has left shifted, and now we're also picking up this long left tail. So we're seeing an increase in heterogeneity in the population with a significant number of cells dividing only a few times at this modestly high temperature. The heterogeneity is even more stark and even more different in the strain on the right, the strongly sensitive strain, where we now see the mode of the distribution at 0. There's lots of cells not dividing at all. But there's still an appreciable number of cells, proportion of cells, that do manage to go through some cell divisions. So even in this very simple application of the technique, we're already starting to get interesting biology out of this analysis. But so far I've only focused on fecundity, divisions. How are we going to get at mortality? So the way we're going to do this is we're going to add a recovery period and a 3rd stain. The logic of this is that wheat germ agglutinin and other bud scar stains can't tell us, or they tend to dye bud scars in cells that are either alive or dead. So we don't know if cells died during their exposure to stress or not. To get at that, if we switch cells to permissive conditions, recovery conditions, and give them sufficient time such that they should have divided at least once during the recovery, if we add a 3rd dye at the end of that recovery period, any cell that divided at least once was viable after the exposure to stress, whereas cells that show no further divisions we'll consider effectively dead. So when we do that, we can further sort of decompose the shifted distributions under stress into condition on whether cells were alive or dead at the end of the experiment. So on the left again, we've got our moderately sensitive strain. We see this interesting pattern where cells that were alive at the end of the stress period, shown in purple, essentially grew as if they were unstressed. And then, of course, there are cells that died. They only went through a modest number of divisions. But here we can sort of chalk up the change in fecundity here to being primarily, change in bulk population growth rate to being primarily driven by an increase in mortality. Those cells that didn't die grew just as well as they did at 30 degrees Celsius. By contrast, in this strain on the right, we have many cells dying, but we also have a change in the distribution of fecundity for the cells that survived. So another advantage of this TrackScar technique is we get additional information for free. We've already gone to the trouble of labeling and counting bud scars, and so we get information about the age structuring of fecundity and mortality immediately when we collect this data, though this is only true for a relatively short proportion of the replicative lifespan of yeast. So we can ask about how did fecundity and mortality change, or what is the pattern of fecundity and mortality conditional on age at the first time point. And when we do so, we get figures like this. I'm showing on the top information about fecundity as a function of replicative age at 30 and 35.5 degrees Celsius. At 30 degrees Celsius, we see no strong structuring with age. If anything, maybe a slight increase in fecundity. And then at 35.5 degrees Celsius, we see this interesting pattern where the moderately sensitive strain shows a slight decrease in fecundity with increases in replicative age. And then, in fact, there's a very strong increase in fecundity with replicative age in our moderately sensitive strain. That makes perhaps more sense when we look at the mortality. We have a very stark pattern of very high young cell mortality in our sensitive strain, which drops very quickly into middle age. If you make it out of childhood, you survive. And then, in fact, a very significant increase in mortality in our moderately sensitive strain, sort of a premature senescence phenotype. Whoops. We've gone after the early cell mortality phenotype here. This very high rate of young cell mortality is reminiscent of mutations that affect mitochondrial inheritance. So we thought that was a likely candidate. And indeed, we've done some work now to show that at 35.5 degrees Celsius, this strain background shows a very high rate of mitochondrial inheritance defects with lots of cells with either no mitochondria or globular, poorly structured mitochondria, a pattern we don't see at 35.5 degrees Celsius in either the moderately sensitive strain or S288C, which is robust. And the mapping work to get at the basis of that is underway. So I hope I've convinced you that going to the extra trouble of decomposing bulk population growth rate into fecundity, mortality, and the age structuring of these parameters is a useful way to gain new insights into cellular physiology and stress responses. And this tool has other applications to the aging and microbial ecology and evolution. With that, I'll thank you and hopefully take some questions.
Paul, that was fantastic. One of the things, of course, you could do with it is to look for persisters. Have you found them?
We haven't gone after the persisters yet, but that's among the things we're hoping to do soon.
So you're not catching the daughters, the newborn. Are they more, but you can tell them because they start with 0 scars and then they might accumulate a few.
Yeah,
are they more robust?
Yeah, we can actually get daughters, right? Because we can get newly born daughters that only have the very large initial bud scar. So, for example, we can tell, it's a very sensitive technique. We can pick up the difference in division rates between newborn daughters and one bud old mothers. So that's something potentially we can look at in more detail, but it requires a lot of...
So are they more robust to temperature?
I'll have to go back and look at the data. I don't know off the top of my head. Great. Thank you.